First of all, let me start by briefly explain the background of our presentation. Our kidney is one of the hardworking organs in the body. The kidneys are two bean-shaped organs that are around the size of a fist. They are placed on each side of our spine, directly below the ribcage, where they carry numerous functions including filtering the blood and removing unwanted and toxic substances into the urine. Apart from that, it also helps the body to stimulate production of red blood cells, control the blood pressure, and balance the body's fluid. However, unhealthy lifestyle and consumption of a diet high in salt or sugar can cause development of diabetes and high blood pressure, which both are significant risk factors for chronic kidney disease (CKD), or also known as chronic renal failure. CKD is an irreversible condition where our kidneys are damaged and loss of function. Because of that, the kidneys are unable to filter out waste from the blood as they should, resulting in buildup of waste and fluid retention. Unmanaged CKD eventually leads to other health complications such as heart disease, anemia, and stroke. The question is, what happens to kidney tissue when it is damaged, and how do we relate the progression of CKD with its histopathology? So, let's hear further explanation from our group about the histology of kidney tissue, histopathology, and its pathological correlation of chronic kidney disease.
The kidney is histologically divided into two regions, which is the inner medulla and the outer cortex. The renal medulla is composed of renal pyramids, and grossly tissue appears striated, while the renal cortex appears more granular. At the junction between the cortex and medulla are millions of functional units called nephrons. Each nephron can be divided into its major parts, which are the renal corpuscle, proximal convoluted tubules, loop of Henle, distal convoluted tubules, and collecting ducts. Renal corpuscle is a location for blood filtration. It consists of the glomerulus as well as surroundings with double-layered epithelial capsule called as Bowman's capsule. Glomerulus is composed of a clump of twisted capillary loops that arise from the efferent arteriole and connect to the efferent arteriole. This arrangement to form a selective filter that will allow certain components of the blood plasma to pass into the Bowman's capsule. The Bowman's capsule has an external parietal layer, which is a simple squamous epithelium, and a visceral layer which clings to the glomerular capillaries, known as a podocyte. The function of podocyte is to prevent the rupture of the glomerular capillaries due to a blood pressure and at the same time allow filtration to proceed. Juxtaglomerular complex includes three populations of cells that help to regulate the rate of filtrate formation and systemic blood pressure. It consists of macula densa cells, granular cells, and extraglomerular mesangial cells. The macula densa cells is a group of tall, closely packed cells in the ascending limb of nephron loop that lies adjacent to granular cells and help in monitor the sodium chloride content of the filtrate entering the distal convoluted tubules. Granular cells act as mechanoreceptors that sense the blood pressure in the efferent arteriole. Extraglomerular mesangial cells may pass the regulatory signals between macula densa and granular cells. The loop of Henle has descending and ascending limbs. The thick descending limbs look similar to the proximal tubule with the apical brush borders, while the thick ascending limbs are composed of cuboidal cells that they do not have apical brush borders. The thick and thin of loop of Henle, also known as thick and thin segments.
The diagram shows proximal convoluted tubule. It receives filtrate from renal corpuscle and responsible for reabsorption of about 75 to 80% of the water, sodium ions, as well as certain sugars and amino acids. The wall of proximal convoluted tubule is composed of simple cuboidal epithelium with eosinophilic granular cytoplasm. The brush border appearance along the lumen of tubule is due to numerous microvilli to increase their surface area. Nucleus is single, spherical, and situated in the middle to basal part of the cell. Next is distal convoluted tubule. It plays a critical role in sodium, potassium, and divalent cation homeostasis. Similar to proximal convoluted tubule, distal convoluted tubule is composed of simple cuboidal epithelium. The main differentiating features of distal convoluted tubule include lighter stay and lack of brush border. Thus, the lumen appears larger compared to proximal convoluted tubule, and nucleus is located close to the apical border of the cell. Collecting duct is the final portion of nephron that transfer urine to the calyx. It can be differentiated from other tubules by the prominent lateral borders of the epithelium cells.
Diabetic nephropathy, or also known as diabetic kidney disease. This disease, known as the leading causes of chronic kidney disease worldwide. It is a manifestation of diabetic microvascular disease involving renal arterioles and glomeruli. There are several stages of histological appearance of diabetic nephropathy, which are early stage, established neuropathy, and late stage. At the early stages of diabetic nephropathy, glucose will combine with amino acid on circulating and tissue protein forming advanced glycosylation end products, AGE. Those products accumulate in the tissue by cross-linking with collagen and modulate changes in cytokine signaling. Accumulations of AGE with collagen and depletion of glycosaminoglycans producing thickening of glomerular basement membrane, GBM. Besides that, we can also observe the mesangial matrix expansion at the early stage of diabetic nephropathy. The glucose promptly increasing the synthesis of collagen and matrix glycoproteins that normally express within the mesangium. Move on to the next stage. When the neuropathy are started to establish, the mesangial matrix continues to accumulate, along with mesangial cells, which resulting in nodular glomerulosclerosis protein called Kimmel-Steele-Wilson nodules, a nodule that comprises collagen and other matrix proteins. High glucose started to causes angiopathy of capillaries in the kidney glomeruli as it decreases the elasticity of blood vessels and causes them to narrow, impeding blood flow, which lead to this nodular formation. Lastly, at the late stage of diabetic nephropathy, segmental and global glomerular sclerosis will appear. The microscopic slide shows the example of global glomerular sclerosis. As you can see, more than 50% of one glomerulus manifested by scarring lesions or hyaline deposition. It is the obliteration of the glomerular capillary lumen by increased collagenous matrix. This feature is a great indication that the patient already in a chronic phase.
Hypertensive nephropathy is another cause of chronic kidney disease. This condition may also be known as benign nephrosclerosis. Histopathological changes of hypertensive nephropathy can be characterized by its vascular changes, which then cause secondary histological changes to other renal structures, including glomerulus, tubules, and interstitial space. There will be two microscopic figures and we will go through them one by one. In the first figure, thickened arterial wall can be seen. This histopathological change can be termed as arterial fibroelastosis. Arterial fibroelastosis is a hypertension-related injury. This change in thickness is due to reduplication of internal elastic lamina with presence of collagen deposition. This leads to narrowing of arterial lumen, thus manifests luminal obstruction to occur. Obliterative vascular changes manifest ischemia of the glomerulus to occur as well as to other renal structures. When ischemia occurs, it causes injury and form lesions to the renal structures. One of the affected structures is glomerulus. A focal globular hyalinized glomerulus can be seen in this figure. Hyalinization is a deposition of amorphous glassy substance called hyaline, which can be seen here deposited in the Bowman space. Green arrow shows a normal functional glomerulus with adequate Bowman space and well-defined capillary loops. Blue arrows show capillaries that are found in the interstitial space. In the second figure, atrophic tubules can be seen scattered throughout the field. Atrophic tubules are shrunken, smaller in size than normal tubules, which is also a result of ischemic injury. PAS, or periodic acid shift stain, is a special stain that is able to highlight the features of basement membrane. In this figure, the tubular basement membrane is observed to be thicker than normal. In the same figure, interstitial fibrosis can also be observed, with accumulation of collagenous material in the interstitial space that is formed to provide structural framework in response to the injury. Arteriolar hyalinosis, also termed as hyaline arteriolosclerosis, is another feature of vascular injury, characterized by irregular deposition of hyaline in the arteriolar lumen.
In conclusion, chronic kidney disease is an irreversible condition in which the kidneys are damaged and not able to filter waste from the blood. As a result, waste will build up to high level in the blood and may lead to other health problems. Functional units of kidney, which is nephron, can be divided into some major parts, which is renal corpuscle, proximal convoluted tubule, loop of Henle, distal convoluted tubule, and collecting duct. In this presentation, histopathologic features of two causes that can lead to chronic kidney disease were discussed, which is diabetic nephropathy and hypertensive nephropathy. By knowing normal histology of the kidney, it will provide a better understanding of the pathogenesis of these diseases.